Hello again, Limassol, Cyprus, the NanoWeek of the European Nano Safety Cluster and the Nano Commons final conference. This is the training day, Thursday, June the 23rd, 2022. And in session 2, we were training on adverse outcome pathways of nanomaterials. The objectives were identification and structure of AOPs, and how to create your own AOP and what formalities do you have to know to submit it. Our trainers in this session were Sarah Paulsen and Ulla Vogel from the National Research Center for Workplace Environment Safety in Denmark.
Thank you. The agenda for this presentation or for this workshop training is to give a short introduction to AOPs. Then I will give an example of how we developed our AOP, to give an example of how to go from research to AOPs. Then I will give an introduction to the AOP wiki where we will browse around. If you have any questions or anything you want to look further at, just raise your hand. And then lastly, I will show you how you can submit an AOP to the AOP wiki, or at least how I did it, because there are different pathways. So this is going to be very down to earth, just basic. What is this AOP? Just briefly going through the structure, as you have seen probably in several different presentations, we have this linear structure going from the molecular initiation event through key events to the adverse outcome. Molecular initiation events are a special type of key event that represents the initial point where the chemical stressor interact at the molecular level with the organism, and that results in a precipitation that starts the AOP. So everything starts with the molecular initiation event in ID. Other big players are the key events. These are changes in biological and physiological states, and these have to be measurable and central to the progress of the defined biological precipitation, leading to the specific adverse outcome. So these are the main building stones, and in the AOP wiki, the moleculation, the MIE and the adverse outcome are also key events. So, but they are more specialized. And just because a particular KE is observed doesn't mean that the precipitation will go all the way to the adverse outcome, but they have to be present for, they have to be preserved for the AOP to go all the way to the adverse outcome. So other players in an AOP is the key event relationships, and these are relationships that connect the key events to each other. They are causal, and they predict the relationship between the upstream and the downstream key events. This means that if you have a measurable key event and then the key event relationships, you should be able to predict the state of the key event coming afterwards. So this is also something to keep in mind when you construct your AOP. Lastly, we have the adverse outcome, which is a special type of key event, and this has to have a regulatory significance and should preferably be accepted, and last line I can see, accepted as a regulatory guideline. So this is the basic structure of the AOP, and which I will be grounded in for the rest of the session. AOPs are singular structures, but they can also be connected in networks, and they can be connected in different ways due to the conversion. The labels are gone, but in the left, in the yellow is the MIE, and then in the right is the adverse outcome in the red. So these are networks, AOP networks connected in the adverse outcome. They can also be connected in the molecular initiation event, and with the yellow circle. So this is another way to connect adverse AOPs together. Lastly, they can also be interconnected on several levels. Here we have one network with several MIEs and also several adverse outcomes. Here we can identify key event nodes that links different EOPs together and provide sort of hubs. And these could provide focus points for example, in vitro assays further analysis. And this was some of the things that I touched upon in my presentation on Tuesday. So this was just a brief introduction to EOPs. And now I want to show how we at our institute went from doing research to construct an EOP. The EOP that we constructed at our institute is EOP 237, which is involved in the secretion of inflammatory cytokines leading to plaque progression. So what's the story, how we ended up here? Just briefly from history and known facts, that inhalation of particles increase the risk of cardiovascular disease. There is a direct link between mortality and concentration of fine particles in the air. And there's this very nice study, old now, where they looked at how the soot in the air was related to heart-related mortality in Dublin after they banned coal for household firing. And what they saw after this ban, which is denoted by the line in the middle, they banned it in 1990, and they could see that the amount of soot was reduced with 70%, but more also interesting was that the level of heart-related mortality was decreased with 10%. Some studies have shown that inhalation of nanomaterials induces the risk of cardiovascular disease. This is shown on the figure here on the right. We have two aortas from mice, and the bottom one is exposed to titanium dioxide, and the top one is exposed to the vehicle. And you can, as you can see in the white spots on the aorta, that these mice develop plaque progression. The thing is that all these things are additive. So you are, if you're living in a polluted area and you're also working with things in your working environment, these things add up. So what we were screening was, could we possibly identify a molecular mechanism behind how nanomaterials induce the risk of cardiovascular disease? So what we did was we took nanomaterial. We used carbon black, titanium dioxide, carbon nanotubes, and graphene oxide, and we instilled mice with these nanomaterials. We then took out the lungs and the livers, and we did microarray on these tissues, and we investigated the global gene expression. We also took out blood to investigate the blood plasma levels. So what we discovered from our gene expression was that all the nanomaterials tested induced acute phase response in the lung, and the most differentially regulated gene was SA3. We saw dose response, and we saw a sustained response. This is a very, very predominant and strong response. We also saw a close correlation between SA3 and inflammation. I'll just find this. This is the figure, the one that should come. So here you see on this graph, you see the number of neutrophils, that's inflammation on the x-axis, and you see the relative SA3 mRNA expression that was in the lung on the y-axis, and there was a nice correlation. So mice have three inducible SA isogenes, SA1, SA2, and SA3. And we also identified levels of plasma protein levels in the blood of the SA1, 2, and 3 after exposure in the lung to nanomaterials. And we also observed a significant correlation between SA3 RNA levels and plasma levels in the blood. So the higher the expression level in the lung, the higher the level of protein in the blood. Yeah, so this was very interesting before. And this is not only in mice, this has also been shown in humans, where after, for example, zinc oxide inhalation in pulp and paper mills and clothing fumes, workers exposed to these materials also have increased SAA plasma levels in their blood. So what is SAA? It's an acute phase response protein. it reacts to it's a systemic response that reacts to acute and chronic inflammation. That conditions that induce acute phase response are also associated with cardiovascular disease, and is traditionally considered a liver response. But interesting with this and our work was we saw that there was actually a very strong pulmonary response, and that this pulmonary response could be attracted to the blood as well. The acute phase response, SAA in functions the way and it has its adverse effects in a way because it interrupts what's called the cholesterol reverse cholesterol transport system. So this is the transport of cholesterol from the arterial wall to the left all the way to the liver where it will be excreted in the bile. So this transport is conducted by HDL particles, and these HDL particles is found ApoA1, the small blue dots. And what happens with when SAA is in the blood is that it replaces ApoA1 as the major HDL protein, and then, as you can see here, and then it inhibits its role in the reverse cholesterol transport. So when SAA is bound to HDL, it does not remove the cholesterol from the arterial wall, so it will build up there. It's also been shown that SAA can induce bone cell formation in macrophages. In this study, in vitro study, macrophages were exposed to SAA and then they were turned into bone cells, which are the primary components of the atherosclerotic plaques. It's been shown that overexpression of SA1 protein accelerates plaque progression. This study, mice were infected with lentivirus, so they either did not express SA or expressed a lot, and the mice who expressed the SA had plaque progression. In this study, it's shown that SA specifically also accelerates plaque progression, and in the same study, they tried to knock out all SAA isogenes, and this resulted in reduced plaque progression. So there is a nice correlation between SAA in the blood and increased plaque progression. So, and this is also been shown in epidemiologic studies. So this is a very nice study in nurses where they have shown that the higher level of SAA in the blood will, and this is a prospective epidemiologic study, will give an increased risk of cardiovascular disease. So this is all the basic groundwork and all the things that forms and that we fed into this EP. So this is how how these things fit together. So our AOP 237 starts with the molecular initiation event, which is sensing of stressor by pulmonary cells. As Geoffrey yesterday, I think, talked about that this molecular initiation event is sometimes one of the things that is the most difficult and the least described. And in our case, this is also the case for this AOP. It's largely undetermined exactly what happens, but fortunately, some of the colleagues, Dr. Sturga and his lab, is doing single cell transcriptomics and trying to figure out what happens when the different nanomaterials are introduced to the lab. And when I went through the posters, I actually saw that Carmela Voss had a poster that's number 76. So if you're more interested in that, you could check it out. It should be in the article. The next thing in this AOP is going to the key event 1, which is increased production of pulmonary pro-inflammatory cytokines. This is well known that the nanomaterials induce.
May I ask one thing in that I would also encourage people to ask eventually if you'll come up with an idea right at the moment, right? Because we're in training session. You said molecular initiating events are not so well described. And point out sensing the stressor by pulmonary cells. So what is the really very early or the first initial event that nanomaterial encounters before they get to the pulmonary cell? Or is there a definition? What is the initial event, or how far does initial go?
It's the whole interacting with the cells, and we still don't know which cells.
Before the nanomaterial encounters the cellular surface, it goes into the lung, it encounters the lung-like fluid.
Yeah,
Is
yeah.
this then the pre-initial, or is the initial then just the pulmonary cell, or is actually the encounter with the lung-like fluid the initial one? And then the next one is then already the event one, or?
Yeah. I think we have colleagues who look at the interaction with the lung surfactant, and we see that for some chronic or some acute effects, acute toxicity effects, that interaction is crucial. As far as we can see for the particles, I don't think it is that crucial. We have tried to have we blow particles into the lungs in different vehicles. We're giving the particles different coronas, and we do see a difference in the inflammatory response, but I think also we see inflammatory response. Now, perhaps Tobias will disagree with me, because he would say, Tobias Dugger, that they see that if they have carbon black in lung surfactant, then they don't initiate a response. But I think that if we focus on lung surfactant, then we get distracted from the real thing here, because I think the real thing is the particle interaction with the cells. Yeah.
Okay, so I mean, it's a definition question. Right?
Have you studied also like macrophage interaction and ILA correlation? Because ILA normally is what attracts your cases.
Yeah, probably in a further key event, right?
And then just to add to it, then we can see that for some materials, the macrophages will just engulf them and not react, whereas for others, for LPS, the macrophage makes the whole thing. They are really, really active. But for some of the others, maybe it's the other cells that actually do whatever information we need. Yeah. So it differs a little bit for the different particles, and and that's why we think that maybe for here, maybe it's not really the initiating event. Maybe it's more the next step where we have.
But where you have the first kind of decision.
Yeah.
It also depends to a certain extent on the pre-existing state. So you described two different scenarios where you have already, I mean, LPS is of course a booster. If you have a kind of a pre-inflammatory situation, then the cells are in a more responsive state than if you would not have such a situation. So, right? Let's get into further detail for that.
Yes, to take that, for example, we have already seen that the interaction of macrophages with several nanomaterials do not cause all the activation of cytokinin. There is no LPS, but is producing ILA activation even if there is no LPS. So
Is that in vitro or in vivo?
it was extracted like a different from monocyte extracts from blood plasma, from donor patients, and then studied in.
In vitro assays using primary cells,
Yeah.
differentiated. But now I guess I have to complete.
Yes. So these key events, one is the increased production of pulmonary inflammatory cytokines. And we see this in the graph here with the number of things broken down, and then leads us to key event 2, which is increased production of pulmonary SAA. And then we move on. This leads to systematic HDL-SDA information, and this is also what we saw in our studies. Then we have that this leads to increased bone cell formation. So again, and lastly to plaque progression. So this is how we structured our AOP. We also have the increased systemic total cholesterol pool. However, when we want to go further with this, we think that maybe this is not so essential, and it has these dotted lines, which means that it's not as tightly connected as the other ones. So this is how we went from our research into making an AOP. And this is the first step. If you want to make an AOP, is to how to structure your research such that it can be shuffled into a linear pathway. So this was the first part of the agenda, and now I would like to give an introduction to the AOP wiki. And just feel free to ask me any questions if you have any.
I have a question.
Yes, of course.
For me to show. So when you make the AOV, do you also propose like a testing strategy? 'Cause a lot of your work maybe comes from in vivo tests, but do you propose how you would investigate each endpoint again?
Yes, ideally, in the AOP, and this is also what I want to show you, there are many different ways of endorsement. So if you don't think about these things really in the beginning, structure it and make it. Of course, you have to have information. You have to have your causality and all these things, but don't fret about the small things, such as this, because it can be accepted at different levels, and then when it's accepted, you can work on it and develop it and all these things. So if you look into the AOPDD, there's many different levels, and some only have the headings. And there's basically no other information than just the headings and then the AOP. And others are fully fleshed out with weight of evidence, and so it's very broad.
I can just say that in our case here, we have really wanted to make an in vitro assay for SAA3, and the big challenge was that if you look at macrophages, which is the traditional producer, if you give them LPS, they will make SAA in mice. If you give them a carbon nanotube, they don't care. So this was actually why Tobias started with a single cell analysis, because we had to try to figure out who is actually making this SAA. So it turns out that it is different kinds of cells depending on the trigger. So again, we have a lot of different pathways kind of going to the same goal, but yeah, through different pathways.
And always remember that the AOPs are fluent and organic, and you can open them up for citation, you can open them up for other people to investigate and input. So think about it as a community more than you giving the facts. So I want to introduce you to the AOP wiki. The link is just aopwiki.org, and when you enter, it looks like this. So it's very, very simple to navigate. So you don't have to log in to look at AOPs or anything, but if you want to register, you have to log in. So what here I'll give you the green tool first. First is the AOP content. So we have all the AOPs here. You click on here, and you are introduced to all the AOPs, and they are listed here, all those that are accepted. You can sort them by different things, by the ID number, the title, the contact, the status, like we talked about, there's many different statuses, and also you can sort them by the different MIEs and AOs. So, for example, we sort them by ID, and we go down to 200, and you can see there are a lot. And this was probably stupid because you can also search, so I don't know why I'm doing this. Yes. So here is my name, and you can say we see the services that it's under development, and that should not be cited, but it's included in the OCD work plan. And then you can click on it, and you enter this the AOP, and there's also a graphical representation, which is not there yet. But then you impute the background, and then you have the different key events here, summary, and everything that's included you can click on and continue. So if you're like, is this linked to other things, then you can click on the specific, for example, key event, and that will show you all the key events that all the AOPs that this key event is related in.
May I ask one thing? You have just mentioned this is under development and shall not be cited.
Yeah.
What does it exactly mean?
Meaning that people should not people should probably not use it. They shouldn't try to change in it, and they shouldn't. It hasn't been fully endorsed yet. It hasn't been gone through with the evidence, weight of evidence, and such. So still under development. So you shouldn't take it as hard facts yet, because all the different things have not been connected fully yet.
But can we mention in the review article?
Oh, yes.
Yeah.
Yeah, but we cannot use it for regulatory purposes. I think that is it, that you cannot say, these particles, like the OECD endorse the
Yeah.
use of ALPs, but this is not qualified for use in OECD because it's not fully endorsed.
It would be cool to be specified somewhere, because we had this question and we did in the end decide to cite
Okay.
in a review article, but not in OECD. And then the next question is also, are you going at the one certain moment to put it into the new status, not under development, that now it's citable? Or is there a process?
It's a matter of funding. It takes.
But it's also a matter of process that you have to make a very comprehensive description of all the key events and the key event relationships, and then they are reviewed, and then you have to defend and review again, and then the OECD will endorse. Yeah. So Sabina Halapanova is going through the process now for the 173 fibrosis AOP, and it was 100 pages of comments and replies. So yeah.
It takes a lot of time to get to that level. Yeah, the key event relationships are also listed, and all these you have to fill in when you construct the AOP.
So when you construct this, did you look at key events that are already in the AOP wiki and what they suit your purpose?
Yeah, and this is
Because I saw you made your own, right?
Yeah. I did, but it's because there was, I of course looked, because that's a very key thing, and also one thing I would add in the end when you are constructing your own is that try to use already present key events. And when I did the network for my presentation and for my work in Harmless, I really saw that there are many similar key events out there that should have been, where people should, instead of constructing their own, should have just taken the old ones, and that would have made a lot of progress easier. Yeah. Then there are different point views of all the AOPs, and then there are also the stresses that you have to come up with when you construct your AOP. You have to mention which stressors will start the AOP. So this is what we have done here. You just remember that the AOP just cannot be stressor. It's not related to the stressor. You have to put them in, but this AOP is not fixed on one specific stressor. So they have to be more broadly. They're not stressor specific. And then you also have to enter a lot of other stuff. But here you see the overall assessment of the AOP, and this is why our status of our AOP is the way it is because we haven't done all these overall assessments, all this weight of evidence and everything to get to fully endorsed. But don't be discouraged. The most important thing is to get your work out there, and then everything can follow later. Just to briefly give you one that is fully endorsed, we can take this one. And here you can see if we scroll down to the overall assessment. So here it's listed, and they have its essentiality, and yeah, so it's a lot of work to get it fully endorsed, and it takes a lot of time. Yes. You can also, the same as with the AOPs, you can also search by key events if you're interested in key events. You can search by the key event relationships and also the stressors. So this is what I did when we did the networks. I searched through the stressors to see if there were anything that matched in the nanomaterials. You can search here on the top with the same with the APs and the key events, and you can also search by ID. So before I could just have searched on 237. So this is the main how to navigate the AOP. It's super easy to understand, and you can get lost and you have fun a lot of interesting things. For the blue tour to get information, here if you want just general information, you can click here, get started here, and it will show you a lot of the AOP principles that is not specific as we talked about. There's modular, all these things. It's very well described. All these things that could be interesting if you want to understand AOP and also make your own. Yeah. Does anybody have any questions for the? Yeah.
So yeah, so when you're starting your AOP, you need to submit it to OECD or?
Yeah, I'll go. This is the next step. So how to so so fine.
And you said that it is open to the community. So if anyone wants to add something, or
No, mine is closed. There are different levels and where they are. And people can always contact me because the contract addresses and stuff is there. So if they have any notes or anything, you're able to contact people, but you cannot write in it or make suggestions.
and for example, if someone tries a new stressor and they learn that it follows the same pathway, then they can add like that stressor that way.
But for our AOP, they would have to write to me and say, I've discovered this. I think this would be suitable, and then it would be very welcome. It's a very kind and open community.
So you mentioned that the development of AOP is a lot of work and this is a lot of time consuming tasks like that. Is there any timeline or like a process that you are going to take to make your AOP and like getting into OECD or like some way of
I think that as my or just in general.
just in general for this AOP, is there a plan?
For my AOP, no, it's about funding, and I created this AOP in a project, and this project it ended, so I would need to apply for the money to be able to do it.
There's a status you can add in the AOP wiki, which is like up for adoption or something.
Yeah.
This will
Oh, so other people can adopt it, others not.
Yeah. This will for such AOPs it will be
Yeah.
easy.
That's
Can
nice.
you repeat the question because
No, it's
was
that
without
he,
my-
he
I'm
mentioned
sorry, I
that,
read that my-
that if you have given up your AOP or you don't have time to go change one or something like that, you can give it up for adoption, actually. So that's pretty nice.
But you say that Sabina has worked very focused on her fibrosis, and that
Yeah.
has taken at least 5 years.
Yeah, 5 years. Yeah. So lastly, and
That's another.
It's more or less follow. I was also interested in the timeline. So you say it's a lot of work and clearly depends on whether it's funded or not. So is it for a full AOP? Is it to get it endorsed? Is it in terms of years or is it really decades?
I think it will be years, but I also think that in the process of making the AOP, you realize that you need to argue for the key event relationships, and they have to be causal, and they have to be quantitative. And I think for a lot of AOPs, you will realize that you actually need to go back into the lab or search for literature or something that will make the process slow. But even if you have the evidence, you have to submit this long story, then it goes into review, and you have to answer. And I think that process takes years,
Okay.
but maybe not decades, but years. And then on top, when you have to fill in all those and the in vitro assays and stuff, because it's for regulation, you really need a lot of evidence.
Yeah, I can see that. Yeah, thanks. But I was just trying to get a bit of grade of time on it.
So how to submit your PVG? There are two ways to do it, and I went through the OECD route. You can also go through the SAAOP route, but I will show you how to do the OECD way. This is it's not that difficult. I've printed out the forms and I'll put it there, and then you can look at it if you want to. But what you can do is you can go through this hyperlink, and this is also available through the AOP, and then you can find the form down here. Complete the form to submit an AOP project proposal, and there's also here a user handbook for AOP development that is a must read before you do it, because then you are certain you're doing it the right way, and also a guidance document. So fill in this and submit it, and then twice a year the OECD members are meeting up, and there will be a slot review where you have to defend or talk about your AOP. It doesn't take that long, and then if they agree, then they will allow you to get access to the AOP to build it up and to write the things down in the AOP wiki, and that's it. Yeah, but I have some notes for you for if you want to do this. So when you construct your AOP, just important things to consider here at the end of this training session. That, as we talked about, it should not be related to nano. It can be a stressor, but it cannot rely on nano. And as was mentioned, you should use key events that exist already that are possible. Yeah. And you should also make key events and key event relationship as should be described as discrete units without reference to specific molecular organization event at the AOs or other key events, which means that your key event has to be there, it has to be measurable and not be dependent. It can be dependent because it's in the EOP, but we cannot link two key events together. They have to be separate units. You should try to include at least one key event on each major level of biological organization. So on the molecular, cellular, tissue, organic, and individual level. And you should favor key events that are more routinely measured and be specific. And then a good colleague of mine, Kenny Newmark, told me that there's been more focus on key event components. So these are also something to consider. They are important because they make AOPs interoperable as they connect AOPs with key events to AOP and key events to ontologies, which are useful for bioinformatic analysis. And this is where it can be found in the wiki. So this is also something to consider, and not something I have considered when I constructed my AOP 4 years ago.
Are you planning to go back?
To my AOP? If I have the time and money. Right now, it's Magnus is taking everything. So yeah, but I would like to. I would like to.
Yeah.
Yeah.
We would love to.
Yeah. And then she also told me, Benny, that there will be an update to the whole AOP wiki in July. So it will include links to third-party tools as well, which is also something to consider. So it can be linked to wiki pathways, for example, and make it all more interoperable with bioinformatics. So that was this kind of session. Thank you.
Thanks a lot, Sarah. Thanks a lot for the questions. But we are not fully at the. Yeah, so we can have some.
And this was a kind of practical question, not the conceptual thing. As someone who's very good in trying to find what methods people have used, understand and if their pathways made up of evidence from various principles. Is there a place where you can write in on there where the evidence comes from, or what method?
Yes, there is, but it has to be in the description. So
Okay.
it's not like you can search on this level yet.
Okay, yeah, I mean, that would be really
Yeah.
helpful.
No, but it's more,
I didn't know if I was missing it.
no,
More questions? maybe some thought also from my side while I'm walking there. I mean, it could be an incentive for researchers if maybe they do not have the intent really to get it to the OECD level because it takes a while to discuss. But it could be an incentive to just deposit an AOP if we have this idea and find something which has not been explored yet, because it's an also entering a kind of a scientific discourse process. Would you agree with that?
Yeah, and also because if that would enable even more links to be connected in between AOPs. So the more information is in there, the bigger and better picture we can get and link them together.
Hi, this is Roland here. I really like that you talked about coupling the description to ontologies. So if you really look, of course, through all the AUPs, it's really a wide west. And you know, when we talk about it, like analyzing a fruit basket,
yeah,
where everybody says, you know, you're comparing, can't compare an apple to banana, etc. You know, we've had this discussion, you know, bring them up here. So are there any efforts somewhere to curate older AUPs which are not really ontology coupled? And is that recommendation your recommendation that you're now saying that people should do this? So how extensive is that? This is an absolute need for us when we want to work broader, have AUPs and all that future, that it's really founded in the ontology. Maybe you have this discussion, I can relate, but
no, it's really good. But thank you, Ronan. What really made me happy was coming to this conference and seeing that different groups are actually working on taking the next step. And as you mentioned, working with the ontologies. And I see different approaches, like your approach, for example, and also from the developers group. I know you're working with it as well. And I think it's very, very nice and very essential.
Yeah, sure. Maybe you have also seen one of the posters from our group where we are kind of proposing this new concept of modified key events by pre-existing conditions of people. So like thinking of vulnerable groups or of, I mean, we're always addressing our models, these normal models, you know, but there are people that have sensitizations going on, chronic inflammatory processes, and so in such way, kind of key events might be modified in a way, or maybe new key events added, which is of course something that is obviously also now it's happening with ontology and or in these kind of subgroup events, right?
Yeah.
You know, I'm very interested in my group in collaboration with you and others to do programmatic. And if we do, I think it's nice to share with the group here. You, for example, look at transcriptomics and the ontology, gene ontology. You cover about 50% if you take a tissue, like a liver or lung. You cover about 50% of the gene ontology or the key events with transcriptomics. You cover about 10% of the MIEs. If we think about the discussions going on in the community, there's always this interest in digging in that. How do we get the molecular initiated? So omics, we really are doing okay, but quite poor. And we need lots of more definition and the MIE that
yeah.
Also, if you look at the description of the MIEs, they're horribly non-uniform.
Yeah.
It's like protein binding or something.
Yeah.
So I don't know. Maybe
I completely agree.
Yeah.
I completely agree.
This is where the community needs to take the next step, and big efforts are needed.
Yeah, Roland is quite correct. We discussed it to a certain extent, the definition of the molecular initiated event, because there are really different understandings, and there are already some models behind, and we have also our understanding, and obviously it's really not a dispute, but a discourse, I would say at least, and we need to maybe agree upon some definition or maybe have some subgroups there too.
Yeah, but maybe, I mean, as researchers, sometimes we are very literate, so we think now we have to see the very first interaction between particle and biology, but maybe what we need there is the first critical event. So which one really matters here?
Mm-hmm.
And I would say for these ones, it is whatever will initiate the inflammation. Not all the benign things that they met something and they were passed on or blah blah blah. Yeah.
Yeah, this actually also ties on this modeling session that we had the other day over there, where we were discussing at the moment, and it was actually Beck who was also active in that. And then a number of other questions went into this direction. I mean, an initiating event in the material bio interaction shall be probably something which somehow alters your proteins because it somehow then displays hydrophobic patches, which could be dangerous signals for the immune system and then would be initiating. So it is initiating, although you might even in vivo model not yet identified it.
So maybe we should think trigger event, or I don't know. I think we are running out of time. So for
Yeah, thanks for
nanoharmony
this
to have time.
interactivity. We were starting a few minutes later, therefore we also
Okay.
gave the time.
Yeah.
Thanks for all that.
And again, the forehand is there up there if you want to take a look at what is required just to get started.
Okay, thank you both once again, Ulla and Sarah, and you are around, of course, as we are all around in this wonderful meeting, in this wonderful setting.
Thank you for your interest in our training on behalf of the Nano Commons project team and the European Union Nano Safety Cluster. Have a nice day. Thank you